And we're live. I would like to welcome all my viewers, I don't know how many that there are, to our final presentation of a tele-visit from a scientist to my seminar and scholarly activities, 165, Symbiosis and Parasitism, in and around us. In other words, never really alone. And today it's my distinct pleasure, because one of the books that I assigned to this entire class was Follow Your Gut, based on the TED Talk by Rob Knight. And I'm delighted that Dr. Daniel McDonald is going to be joining us today. I wanted to change your name as I kept saying it, so I wanted to make sure I had it right. And what I like is, in fact, on Daniel's website, and this is my very favorite part, he begins, "My name is Daniel McDonald and I study poop." And this is just fabulous beyond words. And a former research student of mine, Jill Waters, who now works for Ruth Lay, always wanted to be a poop scientist. So Dr. McDonald, you are living the dream. And so right now, Dr. McDonald is working in the Price Lab at the Institute for Systems Biology up in Seattle. And he joins us today to discuss some of your questions. So I'm going to turn things over to Dr. McDonald. And here is my delightful class.
Great. Well, thank you, Mark, for the very kind introduction. Studying poop has been an enlightening experience over the last couple of years. And Jill's work actually is, that she's done has been quite fantastic to see out in Ruth Lay's lab and linking it back to the human genetics. How's everybody doing today? Yeah. Are you guys enjoying all of these conversations with scientists in the field? Yeah. Cool. So I guess at this point you guys are approaching a fall break. Does that mean that your term papers are due now, or they're due in a couple weeks? Okay, some mix of comments there.
And we have several people who are using aspects of the microbiome as part of their papers, like JT, what's your topic?
Gut-brain axis.
The gut-brain axis. That's kind of the other end of the gut issue, right?
Cool. Well, the gut-brain axis is definitely a very exciting area of research right now. There's, of course, a lot of challenges in studying it because you can't just modify humans however you see fit. So the study designs have to get a little bit creative. So right now, a lot of the work is being done in different model systems like mice. So I don't know if you guys are aware of, but this has come up in your conversation. There's this really fantastic model of a mouse called a gnotobiotic mouse. And what it is a mouse that is raised without any life on or inside of it. And so these mice are very expensive, of course, to produce. And the basic procedure is that you take a late-term mouse that's pregnant, and you give it a cesarean section under sterile conditions, and then you take those pups and raise them in a positive pressure bubble. So they just look like, you know, like the bubble boys from movies or something like that. They just raise them in these positive pressure bubbles, and they feed them only sterile food. But what they can do is, once they have these mice that don't have any life on or inside of them, they can do inoculation experiments and introduce very specific microbial communities. And so they can begin to study things like the gut-brain axis. Like, is there different neurological behaviors that result when a mouse does not have any life on or inside of them? Or if they introduce different microbial communities, does it elicit a different stress response, for instance? And these mice are also the foundation for a lot of the original obesity work that was coming out of Jeff Gordon's group, and also Ruth Lay's notable work from 2006, the lab that Jill is now a part of. So anyways, the gut-brain axis is really exciting. I think that there's a lot of potential directions that can go in. So I had a chance to watch through Seth Bordenstein's talk with you guys yesterday, and that looked pretty exciting. And I also watched through Jack Gilbert's talk, who I know you guys didn't get a chance to interact with, but he did a tele-session last year. And if you ever get a chance to see Jack Gilbert talk, I do recommend Googling him. He's quite a fun individual. But I thought what I'd try to do would be to just go through and at least answer one question from everybody, or try to get the discussion going. And if we make it all the way through, maybe we loop back around and try to knock off the second question from everybody. So the first person on the list is Theo. Theo, are you in the room? Cool. How's it going? So the first question you had was, what's the best way to know what microbes we have on or inside of us? And I might be paraphrasing the question, I apologize. So really, it ultimately depends on the question that you're asking. So if we want to have a rough idea of the composition of the microbial community on or inside of us, we can apply a technique, which I'm sure you guys have heard about quite a bit, called 16S community profiling. And what we're doing in that case there is we are taking a single specimen, say I swab the back of my hand. We're doing some voodoo magic in the wet lab, which ultimately ends up with amplifying a single gene called the 16S ribosomal RNA gene. And that gene is thought to be conserved across all of archaea and bacteria. And then what we're doing is amplifying this gene and doing a whole lot of sequencing and getting a whole lot of short DNA fragments out that give us an idea of the relative abundance of these individual genes. Now, since this gene is thought to exist in all bacteria and archaea, we can use this information to get an idea for how many of a given organism are present within a sample. And there's a lot of hand waviness there, which I'll get on in a second. When I'm talking about composition, this is analogous to, say, taking a picture of a forest and counting up all the different types of trees and plants and animals that are present within the forest. And this is actually quite informative because we can apply formal ecological measures that have been developed over the last 70, 80, 100 years to the study of these small microbial communities. Now, this is quite hand-wavy because if you take a look at just 16S genes, it turns out that some organisms have many copies of those genes. So there's instances of, I think, E. coli that have upwards of 15 different 16S genes. And so when you do this amplification procedure, you're taking a look at all of those different genes that are in those organisms. And of course, because biology likes to, play hell with anything that you want to do rigorously, the 16S genes themselves are not always assured to be identical within a given organism. So there's also instances where those genes from a single organism diverge quite a bit. Evolutionarily. But at the end of the day, it does give us a coarse perspective on the types of organisms that are present. It's not species or strain level of resolution necessarily, but again, just a coarse perspective on what's going on. So this is the type of technique that is very commonly used if you were to read a microbiome paper, I don't know, any of the microbiome papers you guys probably have read are most likely using 16S sequencing to get a rough idea of the organisms that are there. You can go deeper though and take a look at all of the gene content or gene content that is within a sample. And so one of the techniques there is you collect a sample, say from the back of your hand, or a more interesting one like off of toilet paper, and you randomly shear up all of the DNA that's present, and then you try to sequence as much of that as you can. You take all these DNA reads and see what they can recruit to through sequence homology-based methods, like recruit to for a reference database against things that are previously characterized. The disadvantage there's a few of them. One is that DNA sequencing, though it has gotten very cheap, it's still quite expensive if you want to try to get a large perspective on all of the gene content within a sample. So you can do like a back of an envelope calculation on this. If you assume the average genome size of a bacterium is 3 megabases, and say there's 1,000 different bacterium in your sample, you've got quite a bit of DNA just if you wanted to observe like a little snapshot of every single organism, let alone their relative abundances are all different. You really need to sequence quite a bit if you want to get a complete perspective on what's in your sample. The beauty of 16S sequencing is that you only need to target a really small portion of the genome. And so you don't have to do nearly as much sequencing in order to get an idea for what's there. So 16S tends to be a relatively cheap and quick perspective on what's going on in a microbial community. So does that long-winded answer kind of get at how we can begin to take a look at what microbes are on or inside of us? Theo?
Yeah, I think.
Okay.
So remember that they don't have microphones around the tables here, and so that's what we should have in the future. Okay.
Cool. Well, I'll try to judge from your facial expressions whether you guys are falling asleep or So okay, so next up I had a question from Caroline, and the paraphrasing it was essentially the relationship between the microbiome and various different types of mental disorders. And neurological disorders, I should say. And this question came up from a few of you. So the potential for the relationship between on the gut-brain axis, there's a few different avenues where the microbiome may come into play. And I want to preface this by saying that I think that it is unlikely that the microbiome itself might be like solely responsible in contributing to some neurological conditions, but I think more likely it's going to be a case of a mix of host genetics and predisposition, and those predispositions interacting with environmental factors, where your microbiome is a very large environmental factor. So some of the studies that I've found that have actually been that are quite exciting. One in particular goes back to studying mice and in relationship to autism. So there's this really cool mouse model for autism called the murine immune activation model. And this model is really quite interesting because what they do is they don't actually modify the microbiome directly. What you do is you take a mother that's, I don't remember exactly what stage in pregnancy they need to be at, but if you insult their immune system, so if you like inject them with some type of pathogen or something, you know, and give their immune system something to struggle against prior to birth, the pup itself will actually end up with autism-like symptoms as a result. So you're not modifying the microbiome of the pup directly, but you're perturbing the mother prior to birth, and this actually appears to have consequences on the pup that's actually born. So through some work, some building off the murine immune activation model, Elaine Shu and colleagues were able to identify in pups that were perturbed so that they did have these autism-like symptoms, and then pups that were not perturbed so they did not have these autism-like symptoms. They were able to identify a significant difference in the microbial composition of the resulting mice. And from there, they were able to identify a very specific bacterium that appeared to be driving the difference. It was called Bacillus, I think it was Bacillus fragilis. I could be wrong on that. But what they did then was they took it a bit further, and they were able to identify a very specific metabolite, so a byproduct that this bacterium produces, and it's called 4-EPS. And what they found was that the pups that had gone through this, that were part of this murine immune activation model, what they had found was that the blood levels of those resulting pups had an elevation of this molecule called 4-EPS. So what they did was they asked the question, well, if we take this molecule and inject healthy mice, can we induce autism-like symptoms? And the answer to that was yes. And so backtracking, when they actually identified this organism, this Bacillus fragilis, that was producing 4-EPS, they then also asked, well, can we just inoculate mice with this particular organism and induce or that will produce this metabolite, and can we induce autism-like symptoms? And the answer there was also yes. There also, I should preface this, it's entirely possible that I'm getting some of these details mixed up. I do encourage reading the paper because it's been a couple of years since I've taken a look at it, but it is a really fun paper. But the end result was by modulating the microbiome, they were able to induce autism-like symptoms. Now, the thing is, though, is extrapolating this out to humans is really quite a bit of a challenge. For one, humans are, of course, very different than mice, though we do have some relationships. But one of the big differences between humans and mice is the nature of our immune system. And so the microbiome is quite involved in training your immune system, and your immune system can be thought of also as a communication pathway between your gut and your brain. So that we can make this modulation happen in mice does not mean that the same metabolite has the same role in humans, or that the same metabolite might even do anything at all. But what it is suggestive that, you know, this type of behavior can happen in one mammal. So the likelihood that this type of behavior or this interaction with the microbial community can happen in other mammals, I think is likely to be very high. There's been various other bits of work along these lines as well too. I don't remember his first name, but last name of the researcher is McFabe. He's been doing studies involving propionate, which is a short chain fatty acid. And basically you can inject.
Ooh. You froze.
Rats. And then there's also been some work taking a look at notabiotic.
Oh, I think, yeah.
Exposed for, again, mice that do not have any life on or inside of them. If they are genetically predisposed for multiple sclerosis, they will only develop multiple sclerosis if they are inoculated with a microbial community. So it doesn't say that the microbial community is actually what is making the development of multiple sclerosis happen in these mouse models, but it does suggest an interesting link. And again, I think that this is probably a case of some combination of host genetics interacting with environmental factors, but it definitely warrants additional study going forward. There's some really good reviews written by John Cryan. His last name is Cryan. That goes into a lot of detail on the gut-brain microbe axis. And those works, I think, are, if any of you, as some of you are, interested in the gut microbe-brain axis, I do recommend taking a look, or at least searching that individual up on PubMed and seeing what's there. Cool. All right, so Lauren, where are you at? Ah, over in the corner. Okay, I saw your hand. Okay, so should we rethink the idea of a human to include bacteria? So I think that Seth Bordenstein probably answered that question much more comprehensively than I could have. But in my mind, I've tried for the last couple of years to get the phrase microbial overlords put into a publication, but all of my co-authors always seem to take it out at the last minute, or sometimes not the last minute, sometimes much sooner.
You see, Daniel, I wanted to say this is a good topic for me because I am currently sporting a tattoo that reads Awite Pawele Damini, all hail the small masters. And I've been pushing microbial supremacy for some time, so you're in a safe space here.
That's great. That's fantastic. So I think that it would be useful to think of humans as much more than just our human genome, because I can't think of a cognizant human being that doesn't have a very personal relationship with a suite of microbial life. Not just bacteria and archaea, but also fungi and other microbial eukaryotes. And given the correlations that we're observing with health and these organisms, I do think that it is becoming increasingly relevant for the medical community to pay attention to these other organisms that exist. As to how the medical community should pay attention to these organisms, that's where a lot of the research is going right now, is trying to figure out what can we actually derive from these assays that can inform medical decisions. And there's still quite a bit of work to go there. But I think regardless, it would be important and imperative to think of humans as more than just human. Let's see. And actually, Lauren, you had one other question, which I thought was really good. In paraphrasing it, was, do you think that there are problems publishing books like Rob Knight's Follow Your Gut and oversimplifying the research? And I think that it is a slippery slope, because there's quite a bit of hype that can be generated from any type of scientific study, that's in particular studies that are involving human health. And on the one hand, you want to get as much information out to people so that they can learn about all the exciting developments that are going on. And in turn, when the general public is excited about directions that research are going, that feeds back to Congress, and that feeds back to NIH budgets, and that feeds back to researchers' ability to actually go and continue the research forward. So getting out and in contact with the public, I think, is something that all scientists should be excited about doing, because ultimately research is for the benefit of human society. But there's also the benefit of, when the public is excited, it actually enables us to continue forward with research. But we have to be very careful about people mischaracterizing, or just not people in general, but just mischaracterization of the research that's going on. And it's a give and take. I mean, it's a bit bittersweet, but I think that the benefits far outweigh the costs of some research getting potentially mischaracterized or overhyped. And no matter what you do, I mean, people are going to misinterpret things no matter what. So, but I do worry about people making, in particular, uneducated decisions. So I'm very nervous about works that come out that say that there are very specific things that we can do to the microbiome at this point that will, help people. There are some examples, like doing fecal microbiome transplants in the case of Clostridium difficile. There are some examples of the probiotic VSL number 3 being very beneficial in the case of irritable bowel syndrome. But if you go to like Whole Foods and wander, and as Rob has said in his book, I mean, there's a large number of probiotics that you can find at the grocery store. Very few of them have any scientific backing or rigor. And so I think that there is a large amount of concern that people need to take. But in general, I think getting people excited about the research is a very good thing. And hopefully some of you guys will go on and become poop scientists as well too, right? And so, by engaging with the public, hopefully, we can also get the next generation of researchers excited about new and interesting studies. Greg, Carmen, where are you? Ah, there's a hand.
I'll wave again. There you go.
Cool. Okay. So how much of our mood, personality, etc. do microbes control? So this goes back to the gut-brain microbe axis, of course. So that's definitely a really good question, and again, one that's very difficult to study, because you can't just ask a mouse how they're feeling. If you pick them up, they'll probably poop in your hands, which is really beneficial for studying their communities. But we're not able to directly ask how they actually feel. In the case of human beings, we can ask them how they feel and subject them, or if they so choose, to participate in psychological batteries and get an assessment for their overall mood, and then we can ask them to poop on a stick a whole bunch and try to get an idea for what their microbial composition is, or potentially the genes that are present within their microbial communities as well. But because of the total number of variables and the total complexity of the microbiome, an individual's health, as well as their mood, actually identifying this down to these specific organisms are making a person happy, or these specific organisms are making a person sad, is something that would necessitate very large sample sizes in order to be able to tease apart subtle patterns. There's also, we can improve the sample size effect, like reducing the number of samples that we need to collect, the number of participants that we need to collect from, if we do a very good job controlling the study. So what I mean by that is, say, restricting people so that we're only looking within the same age group, or only looking within a very similar BMI, and reduce things that are known to be drivers of microbial diversity. And so then we're reduced in the total variability. But even then, it becomes quite a bit of a challenge to perform a study like this. All this being said, there are a few avenues that I think are that the microbiome definitely can play a role in, if not already playing a role. So the microbiome has the potential to modulate ghrelin, which is one of your hormones that's involved in feeling satiated. Your microbiome also is implicated in the production of a range of short-chain fatty acids. And some of these short-chain fatty acids, such as butyrate, are food for your epithelial cells. If your epithelial cells in your gut and your gastrointestinal tract are not happy, they get inflamed, And when they're inflamed, that can lead to chronic inflammation. Chronic inflammation can impact quite a bit about your body, including something that I just learned relatively recently that I had not realized before. So the same types of proteins that are involved in your in the barrier between your gastrointestinal tract and your bloodstream, they're called, I think, tight juncture proteins. I could be totally wrong on this. My background's entirely computer science, so I'm still trying to get up to speed on biochemistry. But the same type of proteins that are involved in limiting and restricting what is in your gut and getting into your bloodstream are also involved in your blood-brain barrier as well too. And chronic inflammation can potentially make that a more permeable barrier, so that things that are in your blood can cross over to your brain a little bit more effectively. And so if you have chronic inflammation, and if you have things that are leaking into your bloodstream that are derived from your microbiome, there is potentially more of an avenue for them to actually get into your brain itself. So as to if they impact personality specifically, I don't know. You know, are they deciding what we're going to go eat? I think that would be an exciting finding, but I don't know if we're quite there yet. The work again in mouse models, which is where we have most of our understanding of what microbial communities are doing, in particular with neurological disorders, is exciting. But again, translating this to humans is going to be a fun and difficult task. All right. I apologize if I get this name wrong. Please correct me if it's wrong. Braith?
You got it right.
Hey, cool. That's great. So your question was, the first question you had was talking about studies done with mood and microbes and how they're proving it. So that's a really good question. So this, again, goes back to a lot to notobiotic mice. And so one thing you can do is you can take a mouse and say do some genetic knockout and have the mouse be predisposed for anxiety. And you can measure anxiety. So actually the way they measure it, one of the ways they measure anxiety in mice is they have them sit on little platforms that are like, yeah, I think they have them sit on little platforms and they see how long it takes before they get off the platforms. Or they can also have them sit on, like, in stressful situations like platforms above water, where if they fall off, they'll have to swim and there's a threat of drowning. There's a lot of manipulation of mice that are done. But what you can do then is you can assess the stress response in a mouse, say without a microbial community, and then you can inoculate those mice with very specific microbial communities and ask if there is a statistically significant difference in their behavior. And then there's other types of behavior that you could track, like asking how well a mouse is able to find food through a maze, for instance, or other complex tasks. And basically ask at a really coarse level, if we introduce a microbial community, say a microbial community that you would normally expect to find in a mice or in a mouse, is there a behavioral change that you can observe in the mouse? And from there, you can then approximate this out to, well, you know, if we introduce a microbiome and we see a behavioral change, then presumably that means that the microbiome is playing some role within that neurological behavior. But again, if it's like a specific microbe that is eliciting that change, if it's the community, or if it's just that there's a microbial community that is modulating the immune system, because now the immune system has something to pay attention to, but they're potentially all factors. So getting to causation is a very difficult task, regardless of what scientific field you're in. And the amount of evidence that you need really needs to be quite high, as well as ideally having a mechanistic understanding for what's going on. So what I mean by that is having an actual model for these microbes are consuming these bits of food and producing these metabolites, and this is interacting with, say, the gut epithelial layer and maybe modulating the immune system a little bit, and having a complete understanding for what's going on. And that is a bit off. JT, where are you at? Cool. There you go. Yeah. So, can I explain next generation sequencing? I can try.
I'm waiting for this.
My basic understanding of next generation instruments, and there's a lot of different types of next generation instruments. So for, say, the 454 Life Sciences instrument, which is really no longer in use, or the Illumina platform, which is in quite wide use right now, basically they're just really expensive digital cameras. So what you're trying to do, what's actually happening behind the scenes is, and this goes back to Sanger sequencing, is you have some strand of DNA, however you've got that strand of DNA, and then you introduce over what's called a flow cell. You introduce a single type of nucleotide that maybe has some additional groups attached to it that might do some interesting things. So when that nucleotide is incorporated, you can then pause. Sorry, let me step back. Again, I'm not a biochemist. So the basic idea is when these nucleotides are incorporated, they can emit a flash of light. Every single nucleotide emits a different flash of light. And so what the camera is doing is just taking a lot of pictures at a fairly high interval and tracking what different flashes of light correspond to which different portion of the picture. So, some of the nucleotides will flash red, some will flash blue, some will flash green. And from that, you can tease apart what the order and introduction of the individual nucleotides was along a piece of DNA. So a given MySeq run, for instance, will produce 15 million, 25 million individual reads. All of them are on the order of like 250 nucleotides long. So that's a lot of flashes of light, and this will happen in less than 24 hours as well too. So it's total sci-fi that they're able to do this, and is amazing. But that is the basic concept for many of the sequencing instruments that are in play right now, is a lot of flashes of light and taking a picture of those flashes of light. There are some good YouTube videos on it as well too. If you Google like YouTube and next generation sequencing, there's actual images or video of what are called the flow cells. So this is where the individual pieces of DNA are bound, and then they wash over all the different chemicals that need to be used, or reagents that need to be used during the processing and the actual sequencing. But those videos are really cool because you can see all the incorporation of the nucleotides, and you can see the flashes of light, and they'll go into much more detail about how things like Illumina sequencing actually work behind the scenes. So there are some different instruments though, things like Oxford Nanopore and PacBio, and those try to in essence, take a single molecule of DNA and run it over a membrane, and they try to monitor the very subtle electrical changes that happen as every single nucleotide goes through a membrane. In the case of Oxford Nanopore, it's an actual biological pore that they're going through, and I don't recall right now off the top of my head the nature of PacBio. I think they've got some I don't know, polymerase or something like that attached to individual cells within a well, where each well is like nanometers in size or something. The engineering behind the scenes for this stuff is absolutely immense to make it work. So hopefully that was sufficient. If you're ever curious though, you should probably look up mass spec instruments as well too, because they use lasers, and lasers are fun. You basically just blast things with lasers and see what actually comes off of it, and find some fanciful ways to measure those. Mass spec is, I'm only learning about it right now, but it's quite an amazing technology.
There's a related technique, JT, called nanosims. And what they're using is mass spectrometry to find out what's happening inside a bacterial cell, what kind of carbon it's using, what kind of nitrogen. It's fabulous.
Yeah, I mean, yeah, the mass spec stuff it's a growing field, and I think mass spec was first originally developed back in like the 20s or 30s, but they keep expanding that technology, and it's what gives us insight into identifying what individual molecules might be present within a sample. I mean, it's got a very high level of resolution. And this actually, the reason it's important within the microbiome field as well too, is it gives us the potential to listen in on the communication between individual cells and the host, because you can look at those little molecules that are going in between the cell and the host. And that's actually, I think, going to be really important, because ultimately, for many of the questions that we're asking about the microbiome, we don't care as much about what individual organisms are there or necessarily about what the organisms are doing internally. What we care about is what they're doing externally. So what type of molecules are they dumping into the system that might be available for the host to interact with?
So Daniel, when we get toward about a quarter to, we'll start to kind of wind down a little bit so they can get out by 10 minutes to the hour.
Okay, cool. Yeah, I'm not going to make it through everybody. So I had a couple other questions that were marked as that I looked at that I was pretty excited about. So I'm gonna strive for those ones right now. Mary, where are you at? Hey, how's it going? So could our inability to grow many gut microbes in a lab be a big part of why we know so little about microbial communities function and how they interact? And that is absolutely the case. So just like, you're probably pretty comfortable at about 25 degrees Celsius with some relative humidity and access to food, but are probably very unhappy, say, being in Antarctica, totally exposed and without food. Microbial life, there's many organisms have very specific environmental conditions that they subsist in and that they can actually grow and survive in. The challenge, though, is figuring out what those conditions actually are. So if you, as you guys have been doing with your really, really awesome art that you guys have been creating from cultures, some organisms love to grow on agar, but very few organisms actually do. So one of the challenges for the field is enumerating different types of growth conditions and try to expand out a repertoire of what organisms we can actually grow. And this gets really challenging because, E. coli, for instance, can thrive within oxygen, but many of the organisms that live within your gastrointestinal tract live in anaerobic environments. And oxygen is actually quite a reactive species, and not a lot of organisms, or a lot of organisms can tolerate it, but not all organisms can tolerate oxygen. And so in order to try to grow anaerobic organisms, you have to have these very specialized chambers that are called anaerobic chambers. And you can control the gases that are present, and one of them that is not present is oxygen. But one of the challenges also with having an anaerobic chamber is some of the gases that you need to modulate within this environment are quite flammable as well too. And you don't want to blow your lab up, so you have to be very careful about how you actually maintain those chambers. But there's some exciting work going on down at the Joint Genome Institute at Lawrence Berkeley National Labs, where they're using single-cell genomics techniques to try to identify the functional potential of an organism. So they're taking a look at all of the gene content of a given cell, trying to figure out what types of environments or what type of metabolism the organism actually supports, and using that to gear the conditions for trying to grow the organism. So this way it's not a total random search through all possible environmental conditions, which is, I think, effectively infinite. But instead it's more of a guided approach where, this organism appears to like iron, so maybe we should have some iron in the environment, or it appears to metabolize nitrogen, or what have you. But then it actually gets, figuring out the environmental conditions for a single organism is great, but there's likely a lot of organisms that are dependent on byproducts that other organisms are producing. So one in particular that's of interest, I'm not going to remember the name right now, which is unfortunate. So there's some really interesting work going on with a particular organism that its only source of carbon that we can identify is gabapentin, GABA, which is a neurotransmitter that is commonly produced in your gut. It's normally found in your brain. And what they're able to do was what these researchers were able to do was first identify that this organism only appears to grow with GABA. And the way they did this was they had some culture media where if they enriched it with GABA that they had sourced separately, the organism would grow. If they enriched it with all kinds of other neurotransmitters or different sources of carbon, the organism would not grow. So they were able to show that this organism needed GABA to actually survive and grow. But what was really cool was then they took this organism and put it in co-culture with other organisms to identify different organisms that appeared to produce GABA. And they could tell that those other organisms were producing GABA because they had their one organism which consumed it in order to survive. So they were using it effectively as a sensor. But these types of complex relationships where you have one organism that depends on a very specific molecule that is being produced by another one is likely to exist definitely within the gut and in other environments as well too. So it might not be the case of just figuring out how to grow one specific organism. It might actually be easier to grow communities of organisms together and then try to tease out what those individual organisms actually need to survive. And once we can grow them in a lab, then we can poke and prod them and get a better idea of what they actually do as well. Cool. So, and Mary, you also had another question, which I was pretty excited about. So is it possible to use microbes found in nature to develop medicines that combat the negative effects of our overly hygienic lifestyles? So, I mean, we already do this, right? I mean, people eat yogurt and probiotics, and they're already consuming a lot of different microbes on a normal basis, like many people are at least. Whether or not that's combating our overly hygienic lifestyles is a different story, but I think that that's an interesting area of exploration, right? I mean, if you're given antibiotics as a child, does it make sense to then provide that child with a probiotic or say a prebiotic fiber that is, or substrate that is beneficial for some organisms? In terms of using organisms to identify, say, molecules of interest that might have medical relevance, I'm not aware of specific work right now that is doing that, but I strongly suspect that there is a lot of work going in that direction. In particular because there's been a big push to get more mass spec work involved in looking at microbial communities as well too, and seeing what metabolites are produced. But I would not be in a good position to discuss going from observing some molecules that are produced to identifying medical relevance for those molecules. But if you are interested in that question specifically, though, there's a group at UCSD, the Dorstein group, and their focus is on mass spec within microbial communities, and they've been doing a lot of work taking a look at corals as well as samples sourced from humans, and looking at the relationship between microbial composition and the metabolites that are produced. And I think this is a very very exciting direction to go because there are quite a few metabolites that are produced by microorganisms that we have no idea what they are, and the potential for medical relevance or industrial relevance I think is quite high. So to give you an idea, for a given microbial community sample, if you applied untargeted mass spec, so you just try to look at all the different molecules that are coming from this community, you're lucky if you can annotate 1% of those molecules. So we have a very bad idea of like all of the different molecules that are coming off of these communities. And so I think that there's a large amount of room for discovery there.
So Julian Davies has looked into this a fair amount over the years, and I'm proud to say that he was looking for a word for all the low molecular weight molecules being produced all the time, and I gave him a name, the parvome, and from parvus for small, and he actually uses it a lot. And it was nice to have been there at the part of it. This idea of looking at small molecules as effectors for all kinds of things is a very, very big deal, folks.
Julian Davies also, that's the soil professor down at UC Davis, right?
No, he's up at UBC, University of British Columbia.
Is he the one that had the quote that once all microbial life is cataloged, it'll make astronomy look like a pitiful science?
I like that, though, because it's true. First evolved, last extinct, right?
I thought that was it. Sorry, Mary, you had your hand up?
Yeah, what was the group that UCSD called?
Peter Dorstein. The last name is Dorrestein, I think.
You'll never look at a culture the same way.
Yeah, he's done some, one of the recent works was a 3D body paper, which is really cool. They collected skin swabs from 2 individuals at multiple time points, and then took a look at the relationship between microbial communities and the byproducts that were produced. And one of the individuals used a lot of like skin creams and things like that, and that really had quite a profound impact on the types of metabolites that you.
Oh my. They'll be selling that as an aphrodisiac pretty soon. That's
Sure.
coming next.
That's coming. I didn't, that you cut out there, sorry.
Oh yeah, all I was saying is that they'll be selling this kind of stuff as an aphrodisiac soon. Cut out again. I guess it was so shocking, see? Let's see if he comes back.
I'm here, I can hear you now.
Okay, we're back then.
Okay.
You have fabulous video, fabulous compared to the other speakers. I think it's really clear, but you've frozen just a couple of times. I guess that's the trade-off. That or you're messing with us, I can't tell.
Yeah, I don't know. Wi-Fi is always interesting. Do we have time for one more question?
Sure.
Or would you like everybody to? Okay, cool. So let's see. So Colin, you had a question that I was really excited about. So is it possible for an entire community to be wiped out or at least change radically enough to have a large effect on the host? So fecal microbiome transplant is one way to have potentially a large effect on the host. And one of the things that has been observed at least one time, there was one individual who was lean, had a normal BMI, and they developed Clostridium difficile. And they ended up getting a fecal microbiome transplant. They ended up recovering from C. diff, but afterwards they ended up gaining quite a bit of weight. And they, I think, entered either overweight or obese as a result of the fecal microbiome, or likely as a result of the fecal microbiome transplant. So it turns out that the individual that they had received the FMT from was an overweight individual. And so the total change of the microbial community appears to have driven a change in their amount of adipose tissue. But this is also an N of 1, so it could just be, you know, an anecdotal experience. But also in the case of something like C. difficile, your microbial community is essentially already totally wiped out. So fecal samples from C. diff patients basically look like skin samples, to give you an idea.
It's amazing. Yeah.
So, but yeah, so there is at least the potential for this to happen. Whether or not we can control it in a very fine way is going to be exciting to figure out.
Now, at this point, we need to kind of end it. I would like you to do me a personal favor. Several of these students are going to take an exam in intro biology right now. Would you do them the kindness of saying good luck to them?
I wish you guys the best of luck. That will be a fun experience, I imagine.
And see how much they really care about what you have to say. They're not cramming at the last minute. They're listening to you. And so I'd like to have everyone give a big round of applause for Dr. McDonald. And I am so very grateful to have had this chance to meet you virtually in this fashion.
Well, thank you, Mark, for putting this together, and thank you, everybody. This has been fun. There's a lot of really good questions. I apologize for not being able to get to all of them.
Well, you know what? There's going to be a gift coming your way from the class. So thanks again. I'll talk to you soon, Daniel.
Sounds good. Take care.
Bye.